All right, let's just dive right into this ultimate exam review explainer. We're going to cram absolutely everything you need to know about the principles of inheritance and variation so you can completely ace your test. If you're prepping for your genetics exams, you are exactly where you need to be. We're taking all that dense textbook material and breaking it down into exactly what you need to know to maximize your score.
So here's our study blueprint for the session. Essential genetics vocabulary, Mendel's laws and crosses, exceptions to Mendel's rules, chromosomes and sex determination, and finally, key genetic disorders.
All right, Part 1, Essential Genetics Vocabulary. Let's kick things off with a rapid-fire pop quiz on a pair of terms that look incredibly similar but mean completely different things on an exam. What exactly is the difference between a character and a trait?
Okay, so the crucial thing to remember here is that the character is the broad heritable feature. Think eye color or stem height. The trait, on the other hand, is the specific variant of that character you actually see. So blue eyes or brown eyes are the traits that belong to the character of eye color. You've got to keep that hierarchy straight in your head.
Now notice how the prefix homo literally means same. That leads us to a pure line or true breeding organism that carries identical alleles, like a capital T, or lowercase t. It only produces one kind of gamete. But hetero? That means different. A heterozygous organism carries dissimilar alleles, like a capital T and a lowercase t, which means it produces two kinds of gametes in equal proportions.
All right, major exam trap alert here. Please do not mix up your ratios on the test. The phenotype is the physical expression, what the organism actually looks like being tall or dwarf. But the invisible genetic code sitting behind it, that's the genotype, which could be heterozygous TT or homozygous TT. Examiners will absolutely try to trick you by asking for one ratio when you're expecting the other, so watch out for that.
Moving on to Part 2, Mendel's laws and crosses.
You can easily see why Mendel chose the garden pea for his experiments, right? It has 7 sharply distinct, non-blending characters. We're talking things like stem height being strictly tall or dwarf, or seed shape being strictly round or wrinkled. This exact discreteness is what allowed Mendel to calculate incredibly clean numerical ratios and ultimately discover the laws of inheritance.
Seriously, grab a screenshot of this breakdown right now because a super common slip-up on test day is treating the monohybrid ratios as the exact same thing. In an F2 generation monohybrid cross, the phenotypic ratio is 3:1, 3 tall to 1 dwarf. But the genotypic ratio? That's 1 to 2 to 1. 1 true breeding dominant, 2 heterozygous dominant, and 1 true breeding recessive. Oh, and for a dihybrid cross where 2 genes are independently assorting, always look for that famous 9 to 3 to 3 to 1 phenotypic ratio.
Here's another classic test question that literally pops up everywhere. If you have a tall pea plant displaying that dominant phenotype, how do you know if its invisible genotype is a pure capital T or a mixed heterozygous capital T lowercase t?
Well, the answer is you perform a test cross. You cross that unknown tall plant with a homozygous recessive plant, a pure dwarf, because the dwarf plant only gives recessive alleles. If even a single dwarf plant pops up in the offspring, boom, you instantly know the unknown parent was secretly hiding a recessive allele all along. It's kind of like a genius biological cheat code.
Okay, Part 3, Exceptions to Mendel's Rules.
Let's compare incomplete dominance and codominance, because honestly, they're tested against each other all the time. Incomplete dominance creates a totally new blended phenotype, like crossing a red and white snapdragon to get a pink flower. The dominant allele just fails to fully mask the recessive one. Codominance, on the flip side, means both original traits show up loud and proud at the exact same time without any blending. Think of how someone with an AB blood type produces both A and B antigens equally.
Next up, we've got the ultimate vocabulary trap. Examiners love to use this to trip you up. What exactly is the difference between polygenic inheritance and pleiotropy? Okay, here's the ultimate memory trick you need to survive this question. Just think about the relationship, many to one versus one to many. Polygenic means many genes control just one trait. A great example is human skin color, which is determined by the cumulative effect of 3 separate pairs of genes. Pleiotropy is the exact reverse. One single gene affects many different traits. A classic example here is phenylketonuria, or PKU, where just one mutated gene causes reduced hair pigment, skin changes, and mental issues all at once.
All right, part 4, chromosomes and sex determination.
Take a look at this actual past exam question. You absolutely must remember these names. Back in 1902, Walter Sutton and Theodore Bovary noticed that chromosome behavior during cell division perfectly matched Mendel's theoretical factors. They actually united these concepts to propose the chromosomal theory of inheritance, proving that genes physically sit on chromosomes.
So the most crucial point when you're studying sex determination is identifying the heterogametic sex. That's the one that produces two completely different types of gametes. Why? Because the heterogametic sex is the one that ultimately determines the sex of the offspring. In humans and fruit flies, the male is XY. In grasshoppers, the male is XO, which means he's actually missing a whole sex chromosome. But birds, they do things totally differently.
This creates a massive difference between us and birds. In humans, using the XY system, the male sperm carries either an X or a Y. So the father's sperm determines if we get a boy or a girl with 50-50 odds. But in birds, which use the ZW system, the male is the homogametic one, ZZ. It's actually the female bird carrying both Z and W chromosomes whose egg makes the final call on the sex of the baby bird. Pretty wild, right?
Finally, part 5, key genetic disorders.
You've got to memorize these 3 specific chromosomal anomalies, and definitely note exactly which chromosome is added or missing, because they're virtually guaranteed to show up in your multiple choice questions. Down syndrome is caused by an extra copy of chromosome 21. Klinefelter syndrome results in a 47XXY karyotype, which leads to sterile males. And Turner syndrome is caused by the absence of one X chromosome in females, resulting in a 45XO karyotype.
Genes contain the information that is required to express a particular trait. This simple, elegant sentence right from your source material is the absolute ultimate summary of everything we've crammed today. From Mendel's humble garden peas all the way up to complex human chromosomal disorders, it literally all comes down to the information carried as alleles on our chromosomes.
So I'll leave you with this provocative thought to ponder. As we discover more about environmental and cytoplasmic sex determination, are Mendel's basic laws becoming obsolete? Or are they the essential foundation we need for understanding the exceptions? As genetics keeps advancing, we're finding that the exceptions really only make sense because Mendel gave us the rules. Good luck on your exams. Use these concepts as your ultimate study guide, and keep exploring the fascinating biological code that builds us all.